Okay, are there any questions? Close the lecture. Mistake. Why did that not work? I might have trouble with getting my syllabus going now. Oh, it looks like I moved the folder somewhere. What's the name of that? 8552. Can I get that? Nope. Can't copy that, darn it. Having trouble getting my syllabus here, so I'll just go online. It looks like I accidentally moved the folder. Hopefully I didn't delete it. That would be bad. Just download it. Okay, let's share my screen. Looks like the lecture's still working. Nope, don't want to cancel that. There we go. All right, should be seeing the screen now. So today is April 11th, and we're going to be talking about Lab 2, aseptic transfers. Can everybody hear me, and can you see my screen?
Yes.
Thank you. Do you realize you need to read the textbook chapter 3 through the end of the section on the compound light microscopy? You need to have that read before beginning Lab 3, which will start on Thursday. So please read that part of the textbook. Oh, I need to discuss and begin the infectious disease project. Ooh, hopefully I've got that here. Like I said, I moved my spring 2023 folder somewhere. I don't know where. I'll have to look for it. No, that should be a lab project. Let me look here. Infectious disease. There we go. So we're going to talk about the infectious disease project now, and then we'll talk about the lab. It's the grading. It's not the most recent. I'm trying to get the most recent one. It looks like it's this one. All right. The infectious disease research project is a project you're going to work on by yourself, and you'll pick an infectious disease and then do, well, a research project so that you will become an expert on one infectious disease. This project will be worth 100 points. It's a lab project. You can read about the introduction, the rationale, the project format I'll talk about. You need to pick an infectious disease. So it has to be infectious. You have to check with your instructor to make sure that you're the first one to choose that infectious disease, meaning I will not allow two people to do the same infectious disease. Now, there are some ways you can break up an infectious disease. For example, when somebody wants to do the flu, somebody can do influenza A, somebody else can do influenza B, and then a third person can do influenza C. So I do narrow it down so that you have a little bit more opportunity. But like if you want to do Ebola, you have to be the first one to ask for Ebola. All right. Some people have already asked. I told them that I wasn't beginning it until today, and so that I'd let them know today whether or not they're getting the disease of their choice, meaning I wasn't going to start until today, but I'll do that after the lab or maybe even during the lab. You need to check with me for one thing to make certain that no one else has gotten that disease. And then I want to double check that it is actually an infectious disease. Like some people have requested cancer, that's not an infectious disease. I once had a student do it on dental cavities, and I had to say that that infectious disease nature was really, really weak. And that is, yes, a child, when they're born, they will get bacteria in their mouth from other people, largely their parents, although they can get bacteria from different things they're chewing as well, but chewing on, I should say. But the point is that every child gets those bacteria. And so it isn't an infectious disease in the sense that one person can get the cavity and then they can give the cavity to another person, or the bacteria that causes the cavity to another person. But generally speaking, the bacteria we have in our mouth cause the cavities. So I'm not going to allow that ever again, because I was hesitant in the first place, and I decided that after I read it that this is not really an infectious disease. There are a few diseases which aren't communicable, which are considered infectious diseases. And something like tetanus and botulism are considered infectious diseases, but they are not communicable. So there are some diseases like that that you can choose, but generally, to be an infectious disease, it has to be communicable. Like I said, there are exceptions, like tetanus and botulism. Any questions about what you need to do? So you need to check with me, email me about what you would like to do. You have to pick a infectious disease yourself. And if you're one of the early persons, you can send in one disease. But if you're going to wait and send it in at the end of the week, you should probably send in at least two possibilities, because by the end of the week, there will be a lot of students requesting. And if you have two shots of getting a disease, you're more likely to get a disease. And then the other reason why I want to have you request from me is I will narrow it down. Like, you're not going to be able to do the flu, but you could do influenza A. You cannot do pneumonia, but you can do an infectious bacteria or virus or fungi that causes pneumonia. So you take one agent that causes pneumonia and then do your report on that one agent causing pneumonia. Is that clear? So in reality, there's probably 20 different agents that cause pneumonia. So 20 different people could do pneumonia as long as they break it up. Any question about that? All right. So that's what you need to do. Once you pick your disease and it's cleared by me, then do a search of the literature, books, magazines, news media, scientific journals, internet. Make sure you have good sources to learn about your assigned disease. To make sure you have a good source, don't take from a web page which is biased, like a political web page or a religious web page or somebody who's just out there making a certain agenda. Okay, make sure it's a good source. You can initially use Wikipedia, but you cannot use Wikipedia as a reference because anyone can go into Wikipedia and change what's in Wikipedia. But you could certainly do your preliminary research using Wikipedia and then use other research to back up your statements. Is that clear? All right. So this is going to be a written report where you detail the information about your disease. The report must be typed. Of course, it's going to be sent in by emails or something like that online. So it should be single space, but I'm not really going to mark you down for that. Should be 1-inch margins. I'm not going to measure that. I do use, please, 12-inch font, 12-point font. Sorry, 12-point font. If you notice, I wear glasses, and if it's anything smaller than 12 point, I'm going to have a hard time reading it. And you're going to suffer in your grading if I don't want to grade it. So make sure it's 12-point font, okay? Just be kind to me. I've got glasses and my eyesight isn't as good as it used to be. The report should include the following items. It should have a cover, but the cover should not be a cover page. Just put the cover on top of your text. In the cover, you talk about what the title of your report is, your name, I don't know, the class name, whatever else you want. But then immediately underneath that, start your text. So don't have the cover on its own page. It just wastes page. Although I'm probably not going to print it out. If I do, I will definitely not want a cover page. The text must not exceed 5 single-sided pages. If it's less than 4, you will get a penalty. If it's more than about 6, you will get a penalty. And when I'm counting the pages, I'm not going to count pictures and your references. So I'm talking about the text proper. It must describe all aspects of the disease organism that are itemized in the content report. I'll talk about that later. The material of the text is going to be worth 60 points, but there's an additional 15 points for grammar. So make sure your grammar is appropriate and good. Spelling, flow, and readability. So I should say 15 points for grammar, spelling, flow, and readability. And I generally have a spell checker, so, don't try to fool me on your spelling. Flow, just do your best writing it up, and readability. Generally speaking, people are pretty good with grammar, spelling, and flow and readability. The exception is if English is not your primary language, you should probably write it early and then send it to someone who will check the English and make recommendations. And the writing lab at Clark would be an excellent place to send it, but you could send it to someone who's just good in English and they know science, for example. As much as possible, the text should be written in your own words. If it's not written in your own words, you must use direct quotes and only when absolute necessary. I once had a student turn in over 50% of her paper in quotes, and then she cited where she got that information. And I decided I'm not going to ever allow that again, that if you have that much direct quotes, I'm going to tell you that you're not plagiarizing because you quoted it, but I'm not going to allow this. You're going to get a penalty because it should be written in your own words. I don't want you copying and pasting from other places and then putting it in quotes and saying where you got it from. Any question about that? The paper has to be submitted in a specific format, such as Microsoft Word. If you don't have Microsoft Word, submit it as a rich text format. You may also submit it in a few different formats like text. I don't remember all the others. You cannot submit it as PDF. And then the paper will be checked automatically for plagiarism. Okay? Anyone whose plagiarism is above 20%, I will go look into the plagiarism. Okay? And if it's above 30%, you'll probably receive a penalty. If it's above 50%, you'll get a flat out 0, a score of 0. Okay. Now, sometimes if you've got quotes and then your references will be shining up in the plagiarism. So almost nobody has a report that's 0% plagiarism because they're using quotes, even if very seldom, and then their references. That will flag the plagiarism. So like I said, as long as your plagiarism is less than 20%, I'm not going to look at it. If it's above 20%, I'll go look at it. And like I said, if it's above 30%, you're going to start seeing a penalty. Above 50%, you'll get a 0. You should use in-text citations. You use an in-text citation whenever you use a fact that is not commonly known. For example, I cannot tell you how many people have died in the United States from COVID-19, but let's say you give that number, you should give that in, after that number, you should give an in-text citation for where you got those figures. Let me start a new one and show you how to do an in-text citation. An in-text citation is in the text. So blah, 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 that's our text. And then you give a fact, I don't know, COVID-19 in USA is, and you give a number, I don't know, we're just going to go XXX. And then you do the in-text citation where you give the author's name, and that's the author's last name, comma, and then the year. So an example of that would be Smith, in parentheses, Smith, 2000, oops, what happened here? 2023, because obviously you're giving the COVID-19. If it's more than one author, you go Smith and Jones. Use both of their last names and then give the year. And that should be in parentheses. Generally, should be in parentheses. The exception is if you're saying something like And I learned from Smith that the number of people receiving COVID-19 in the United States was some number, and then you can just put it the year in parentheses after Smith. But generally speaking, you'd put after the number or after the sentence Smith, comma 2003, and then period. If it's more than two, you go the first one, Smith, and then you go et al, and then you go the year, which this one I'm going to do a different year. Oops. Any questions about an in-text citation? That is how you do an in-text citation. This is the only place where, well, almost the only place you're allowed to use et al, and that is in the in-text citation. Any question about the in-text citation?
If for some reason there's no author, which I'm sure most of them will, do you want us to put like the title of the article or just the year or?
The MLA tells you how to do an in-text citation. I don't want to state anything because I want it to be correct, and I'm not exactly sure. What you can do is email me that and I will look it up, but I don't remember what you do exactly in the case of an author. If it's a web page, you can usually, where do you go? Oh, that's the wrong one. Here we go. You can usually go to the web page and go, I don't remember how to do this anymore, but go to the source. Anybody see source here? Do you know what I'm talking about? Bring up the source. Oh, good heavens, I don't see any source here. Maybe that's this browser.
I think you might actually have an MLA citation like document like in this general course information. So I can
Yeah,
always
it's
that.
there. I'm just telling you, usually you should go look for the author. Okay. Meaning
Okay.
if the author isn't there, you should be able to find it if you look. Now, if it's something like CDC, a page from the CDC, then there may not be an author. Boy, I'm not finding any source anymore. It used to be something that was very easy to get, and I don't even have the controls here anymore for getting that. So I'm not finding how you get a source. If you can get the source, like I said, that's an easy way to get it. And let me look on a different browser here. Nope, this one doesn't have it either. I guess I'm dated. When I was there, you just went to the menu and you could get the source. And of course, now they don't have the menu. You got to click a button to get the menu. And I don't see the source anywhere. Let's look under more tools. Ah, there it is. Under more tools, I found the page source. So this one is not giving the author's name. However, it's just a blank Mozilla page. Let me go to a page on the web. Let's go. Oh, what would be a good one? Maybe a newspaper. Well, actually, you get a newspaper article, it'll always have the author. So maybe that's a bad one to go to. Like I said, for some internet pages, you won't be able to get an author, but do look. And an easy way of finding the author is to go to the source, page source. If you're using a scientific journal, there will always be an author. Okay. It's only if you use an internet page like something from the CDC, where it's a general page and there's more than one person who wrote the page, it will not give an author. All right. So many pages do have an author, but finding that author is difficult. You got to go look for the author. And the author is the best way to give an in-text citation. Like I said, I don't remember if you don't have an author, what you do. If you do like no author and then state the journal name, which I do not want in an in-text citation because that would be too long. So I don't even remember what you do for an in-text citation. You could give you wouldn't want to give the web page address either because that would be very long too. But like I said, it is stated somewhere in MLA and APA, and I'll have to look it up because I don't remember what you do if there is no author. Sorry, that's the best I can do at the moment. Like I said, send me a question because I'm going to forget. I will forget. So send me an email and I'll go look it up for you. All right.
Sounds good. Thank you.
All right, let's go back to talking about this project. Oh, I do want to keep that up. So in-text citations are worth 10 points. You may use any common citation format. You can use APA or MLA or a science citation format. Just be consistent and only use one format per paper. Sources you cite must be scientifically appropriate and reputable. Government sites, USDA, FDA, CDC, scientific journals like Nature, Science, popular magazines, Scientific American, National Geographic. Books and textbooks are good. All science textbooks, probably all course textbooks, are going to be reviewed by experts in the field. And so by a reputable source, we want that page to be reviewed by experts in the field. You may also use a variety of scientifically reputable television shows or channels like the History Channel, PBS, but be very suspicious of any religious site, political site, or privately funded site because they will have an agenda and they may not be correct, meaning their facts may not be correct. College and university pages that contain student work is not something you should use. Generally, professors are correct, meaning they'll not have false information on their web pages, but their web pages are not reviewed. Okay, so if it's a college professor, their website, you can probably use that information. But if it's a student website, I would not use that information because it might be, the information may not be correct, might be not factual. You should not use personal websites. You shouldn't use Wikipedia only because anyone can go into Wikipedia and then change it. But you may use Wikipedia to start your research. And then when you're using your in-text citation and your formal references, just don't use Wikipedia. Back up your statements in Wikipedia from some other source. You must have a literature citation page after your report, which this is the formal references, where you list all of your in-text citations. However, you put the full citation in the full citation form. Is that clear? At least 4 of the full citations must be unique sources that are referenced in your report, meaning peer reviewed. And I can just generally look at your references and tell whether it will be a peer reviewed reference or whether it is a not peer reviewed reference. Okay, for me, that's very easy to tell. And obviously, you're doing the New York Times. That's not peer reviewed, but it's acceptable because they are factual. They try to be factual. But you do something like the National Enquirer, I'm not even going to allow that. Okay, don't use the National Enquirer because they're not factual. Any questions about any of that? All right. Your literature citation page, meaning your formal references, will be worth 15 points, and it must include the full citation. Your plagiarism tutorial told you how to do a full citation. For example, for Smith, 2003, in the full citation, this is not an in-text citation, but a full citation, you would have Smith, 2023. You then, actually, you should have more than just Smith here. You should also have at least his initials. So we're going to go AJ. 2023. You then give the article name. Oops. And then if you got it from a journal, you give the journal name, the volume number of the journal, and then the page numbers. Article name, journal name. I think that's right. If it's a textbook, it's even more than this. For a textbook, you give the author, the year, the article name, the textbook name, or the book name, I guess we should say. Book name, the book publisher, and before that, you should give the book editor, the book publisher, and then the city of publication. If there's more than one city, you just need to give the first one. Okay. If it's a web page, then you give the article name, and then the web address. If it's a page that can change, you also give the date accessed. And sometimes you give some initials and stuff like that. I don't remember what that's for. Any question about any of that? All right, so that's a full citation. In the full citation, you generally do not use at all. There is one exception, and I'm not going to talk about it. You can read about it in the plagiarism tutorial. There is one exception where you can use an at all in giving a full citation, and that's when you're listing 7 authors and there's continuing, then you can go at all. So you don't need to go all of them, but you must give the last author if you go at all. Any question? And you have to give the first 6 authors. All right. So that says in-text citation, a full citation. Do this right. Okay, students are often marked down on either the in-text citation or the full citation or both. So do that right. You must submit the report to the folder in Canvas, and it'll be the project name. It'll be under the lab, so the week of the lab. The project content should have headings in your paper so that I know where each section begins. So you should have the article name and then the causative agent with the heading. And then you talk about the causative agent. I'll come back and talk about that. And then you have the next heading, history, talk about the history, epidemiology, talk about epidemiology, etc. And then talk about pathology, response and treatment, and then the social, political, economic considerations. Okay? If you don't have those headings, you'll be marked down 6 points because there's 6 different headings. The causative agent, it depends if it's a bacterium or a virus or a prion or a eukaryote like a fungus. And you have to state specific information about what the agent is. What's the causative agent? If it's a bacterium, you should say some characteristics of the cell, whether it's gram positive or gram negative, its cell shape, the cellular growth arrangement, some metabolic capabilities, and anything unique or special about that organism. If it's a virus, you should state whether it's a DNA virus or an RNA virus, whether it's positive strand or negative strand RNA virus, whether it's a retrovirus, whether the virus is enveloped or naked. You don't know much about that, but we'll talk about that later in the term. How large the virus is. Does the virus have a provirus? Are there any unique viral characteristics or multiplication cycles? Okay. And then talk about the history. How long have we known about this disease? Historically, how was this disease treated? What's the history of the vaccine development of this disease? Describe the changes in our knowledge and attitude towards this disease throughout history. Even COVID-19, we've changed our knowledge. Obviously, we know more about COVID-19 now, and our attitude. Like, I go into the store all the time wearing my mask to make sure I don't get COVID-19. And most of the other people in the store are not wearing a mask. So our attitudes have changed over time. Even though COVID-19 has been in the United States only since 2020. It's been in the world since 2019. Talk about the epidemiology, describe the prevalence and transmission of the disease, where in the world the disease is found, how many people are currently infected, what's the rate of new infections, what the rates of morbidity and mortality. Mortality is the death rate. What are the reservoirs of the pathogen? Where does it come from? And then what's the mode of transmission? Pathology, describe the pathogenic effects of the disease on cells, tissues, and organ systems. And so we're talking about if you have a bacterial infection, how the bacteria, what is the bacteria's effect on cells, tissues, and or organ systems? What tissues, cells, etc., does the pathogen affect? Like, for example, polio affects the nervous tissue, specifically the nerves of the spinal cord. Damages the nerves of the spinal cord. Is a toxin produced by the organism? Does the immune response cause damage? What is the time sequence of disease? Meaning, what's the length of incubation? What's the prodrome period, the illness period, the period of decline? We'll talk about all these when we're talking about disease. And then what are the major signs and symptoms of the disease? Like everybody knows with the cold, your major signs are a sore throat and you have a runny nose. You have other signs and symptoms too, but those are the start. Response and treatment describe the activity of our immune system against the pathogen. Is the immune system cell-mediated immunity or humoral immunity? That means antibodies. Is it innate immunity? That's what children use to fight off COVID-19, or is it acquired immunity? This is mostly what adults use when they've been immunized against COVID-19, meaning adults don't have a very good innate immunity against COVID-19. How is the disease diagnosed? What types of medical treatments exist? Describe these treatments and the effect on the progression of the disease. Are there any antibiotic resistant strains? Describe prophylactic measures that can be taken to limit the risk of infection. For example, when I go to the store, I wear a mask. And the reason why I'm so protective is I'm not around students, I'm online. So I have very low exposure to infective agents, including COVID-19. And then talk about the social, political, and economic considerations, and you can read about those. Any question about that? I have a grading rubric found on the next page, and this is how you will be graded. You'll be given 1 page for your cover information, 1 point, for your cover information. You'll be given 9 points for the causative agent. If it's bacterial, you need to give the gram reaction, the shape, the cellular arrangement, any metabolic capabilities, and then unique features about the causative agent. On the history, it'll be worth 10 points. Epidemiology, 10 points. Pathology, 10 points. Response and treatment, 10 points. Social, political, and economic issues, 10 points. And this one will be easy. And then you'll be at 15 points on the quality of writing. So what's the grammar, the spelling, the punctuation, the flow, and the readability? Generally, unless you do a really poor job, people only will get a -2 for that. In-text citations, correctly citing information that is not common knowledge. If you state things that I do not know, and I know a lot more than a lot of the general public for a lot of these diseases, if you state something that I don't know and you don't have an in-text citation, I'm going to know about it. And if you do that more than just a couple of times, you're going to get a penalty. Okay, so you do need to back up your statements. And then if your format is incorrect, you will also get penalties. Literature citation page, the formal formats are worth 15 points. You have to give full references for all your in-text citations. At least 4 of the references must be from reputable sources. And that'll be a total of 100 points. Any question about the grading or the project? All right, that's your infectious disease. So let's talk about lab 2, and I'll try and be quick about this. Hopefully I didn't move that one. There we go. Oh, didn't get it. Let me shut down the syllabus. I don't want too many things open. Actually, let's check and see if this is finished. It is, good. I can shut that down now. All right. Lab 2, this looks like the worksheet. Lab module 2 on aseptic transfers. The primary learning objectives are upon successful completion of the lab, you should be able to understand the purpose of using aseptic transfer techniques, determine if aseptic transfer was used from the data provided, and be able to define the following terms: ubiquitous, aseptic transfer, inoculation, and contaminant. In our local environments, microorganisms are ubiquitous. This is a problem when working in a microbiology lab because we generally want to work with a pure culture, a culture containing only one species of microorganism. Louis Pasteur developed a technique of aseptic transfers to minimize the risk of contamination. Louis Pasteur also invented the technique for getting a pure culture. We will learn how to make aseptic transfers in this lab. During inoculation, never lay the test tube cap down on the workbench or the lid of a petri dish down on the workbench. And you shouldn't take the lid off. So this is the lid, okay? And then put it upside down, which that actually is better to do upside down than put it down. I don't know if you guys can see this. Put it down on the workbench that way, because you put it down on the workbench that way, you'll definitely get contaminants on the edge of the lid. The danger of doing this is if you put your hand over the upside down lid, then things will fall down from your hand, and they may fall down from the air. But if you did that briefly, like within 20 seconds, you'd probably be okay. To minimize contamination, you want to tilt an end cap tube and then keep the plate partially covered with the lid. And this way, if bacteria were to come down, most of the bacteria falling down will hit the side of the tube and will not get in the media. There is obviously some risk, but only bacteria coming directly here. And that's less risk than if you were to have the tube straight up and down. Any question about that? The same with the lid. If bacteria were coming down, falling down from the air, most of it will hit the lid of the plate. Only the bacteria falling here would actually fall within the plate. And that's to minimize the risk of contamination. The other thing you want to do to minimize the risk of contamination is to work quickly, meaning have this tube uncapped for the least amount of time you can. Have this plate lid open for the least amount of time you can. So work quickly, work aseptically, and then cap it so you have minimum contamination. Let me show you something. This is barely showing it to you. Oh, I need something to stick inside. Not exactly the best thing, but you'll get it. Hopefully you can see this. We're going to pretend this is my tube. And when you're putting let's do another one that's even better. Here we go. Hopefully you can see this. Can you guys see the pen and the dish here? Anyone? Can you see the pen and the dish?
Yeah.
Okay. All right. So the loop, you notice, has a metal portion and then a handle portion, which is not metal, That's usually rubber, so that when you heat this metal portion, it doesn't burn your hand. The heat doesn't transfer to your hand. Well, the loop portion, the wire portion, can be sterilized by getting it hot. The rubber portion cannot be. And so I'm going to pretend that the green part is the metal part, and then this is the rubber part. What you want to do is stick only the wire portion in the tube or the plate, and don't stick the rubber part in the tube. So they have it shown where the rubber part isn't going into the tube, but just barely. And my point is just make sure only the sterile part of the loop goes in the tube. And if you need to bring the fluid up to touch where the bacteria is on the loop here, you tilt the tube so the fluid moves towards the loop. Don't send the loop further down into the tube, because if you do, you can risk bringing in bacteria and organisms on the rubber part of the handle into the tube. Is that clear?
Yeah.
Okay, great. Let me throw that back away. So in order to inoculate your tubes and your plates, that is transfer cells into, you put it into new sterile media. And the media has been sterilized by putting it in a autoclave, which is a giant pressure cooker. And we'll talk more about an autoclave later. So everything is sterilized that can potentially contaminate the new sterile media. And the media we're going to, here it is here, the sterile media, we're going to put the cells from the stock culture in. This is sterile. And then we sterilize the loop either in a Bunsen burner or in the Bacto incinerator. And then we put the loop into the stock culture, cool the loop, and then put it into the culture, and then take the cells out of this culture, cap that tube, take the cap off of this one, and then put the loop into this tube. So everything is sterile beforehand. And there is an actual inoculating loop where you can sterilize this portion, the wire portion of the loop up here, and then the wire portion. You don't sterilize the metal portion, which is the part of the handle, or the rubber portion of the handle. All right, know the terms of this lab. And then in lab number 2, you're not physically going to be performing any of these activities, but you will be watching videos of these activities. And we expect that you will know and understand the material in these videos as well as in this lab. In procedure 1, you're going to be transferring a sterile liquid using a glass serological pipette. And we have a little video here demonstrating proper aseptic transfer. So watch that video. I'll come back to procedure 2. In procedure 1, you're going to be using aseptic technique to transfer 1 ml of sterile water into a tube of nutrient broth. Now, when you're measuring water, and actually anything in a pipette or in a cylinder, the liquid will not go across flat. It'll form a meniscus and look like that. And where do you read the meniscus? Do you read it at the top of the meniscus, the bottom of the meniscus, or in the middle? Anyone remember from your previous class? Nope. You read it at the bottom of the meniscus. So that would be 1 mil where the bottom of that is. You appropriately label the tubes with your name, the date, the nutrient broth, and then what you're going to grow in the tube. And then this gives you the procedure for performing aseptic technique. And then in procedure 1, scrolling down to the table down here, you use this table to interpret the results. You're not going to be actually making these observations or doing any of this in a lab because it's all going to be done online. But we will be giving you results and pictures, and you will be able to answer the questions from the results and the pictures. So, excuse me, for procedure 1, if the nutrient broth is clear, then no organisms are growing in the broth and aseptic transfer was successful. So that's the result. This is the conclusion. If the nutrient broth is turbid, meaning hazy or cloudy, organisms are growing in the broth and aseptic transfer was unsuccessful. So you use this table to determine your results and then make your conclusions. Any question about any of that? Now the results are given in this picture here. This picture here is for procedure 1. This picture here is for procedure 2. That picture here is for procedure 3. And this picture here is for procedure 4. You have to remember what you're doing in each procedure to be able to answer these questions. So describe the initial clarity of the nutrient broth prior to inoculating, adding the sterile water or the paper disc. And I think on your copy, I've got this labeled 0. Let me look at that. Actually, I probably have it in the worksheet. Well, let me look here. I need to update my teacher's copy. Now, let's see if it's that way online. I thought that was labeled. We don't have that label. That's still a 0 there. It's not a 0 there. But you need to answer this question. Describe the initial clarity of the nutrient broth prior to inoculating. And we have a picture for that. Let's see where that is. Come on. Oops, wrong one. And there it is there. So tell me whether that's turbid or cloudy. And then for the result for picture 1, describe the clarity of the nutrient broth after inoculating with sterile water and overnight incubation. Tell me whether it's clear or turbid. Based on these results, what conclusion can be drawn? So you take a look here, go up to the table, and give me the conclusion. The conclusion must be worded similar to the conclusion in the table. If it is not similar, I will tell you to do it over again and use the wording in the table, meaning if the nutrient broth is clear, you make a statement, no organisms are growing in the broth, aseptic transfer was successful. Okay? Any question about any of that? And then list 3 sources of error with aseptic transfer technique that could explain why the nutrient broth showed growth of microbes. So 3 sources of error in the technique. Okay, any question about any of that? If not, let's go on to procedure 2. Procedure 2, you're transferring a sterile paper disc using sterile forceps, or at least clean forceps, into a tube of media. That doesn't state there, but I know. It stated up there. Nope. And this is the procedure you're using in procedure 2. You're not going to actually do it, but you'll pretend to do it. So you should read the procedure. If the nutrient broth is clear, then here's your conclusion. If the nutrient broth is turbid, hazy, or cloudy, then here's your conclusion. Any question about that? So 2 goes to procedure 2. P stands for procedure 2. Describe the clarity of the nutrient broth after inoculating with sterile paper disc and overnight incubation at 35 degrees. Was the tube clear or turbid? And that will be P2 right here. And then based on these results, what conclusion can be drawn? Once again, use the table to write your conclusion. All right, let's take a look at P3. I probably went over, but let's make sure. Yep, I did. Procedure 3, you're going to transfer Serratia marcescens to a broth. For procedures 3 and 4, please watch the following video demonstrating how to correctly transfer from a bacterial stock culture to a sterile broth and to a sterile agar slant. You only need to watch this video to 4 minutes and 23 seconds. You might notice an error made by the instructor filming this, and he puts the loop into the E. coli stock tube. You hear something, and you know what the error is. And you can hear sizzling when he puts the loop into the E. coli. And that sizzling is around 3 minutes and 5 seconds to 3 minutes and 20 seconds in the video. So in procedure 3, you're using aseptic technique to transfer Serratia marcescens to a sterile broth culture. Serratia marcescens, when it grows in a liquid culture, is pink. When it grows on a solid culture, it is red. So this bacteria is pigmented. Broth cultures are problematic because it is difficult to determine if contamination has occurred. So if we inoculate this tube with Serratia marcescens, it would be difficult to determine if we had minor contamination because all the cells mix together and it will look pinkish in color. It's only if you have a major contamination would you notice contamination in this tube, meaning if you inoculated it with Serratia marcescens and the growth is not pink or pinkish, and it's some other color like white, then you say, oh, I have a major contamination here because the color is not correct. Any questions about any of that? Anyways, there's the procedure for working with a broth culture. You use this part of the table for procedure 3. If the nutrient broth is clear, then the conclusion is no organisms are growing in the broth. Aseptic transfer was unsuccessful. The reason why it's unsuccessful is we put into tube 3, we specifically put Serratia marcens. So we do expect organisms to grow in the tube. For procedure 3, it also states whether the nutrient broth is turbid. If it is turbid, then the organisms are growing in the broth and aseptic transfer was successful. As long as the growth is pink, it was successful. All right, let's go down to procedure 3. Describe the clarity of the nutrient broth after inoculating with Serratia marcens and incubating overnight at 30 degrees. Was it clear or turbid? Based on these results, what conclusion can be drawn? So take your results here, clear or turbid, go to the table. If it's clear, here's the conclusions. If it's hazy or cloudy, this is the conclusions. Oops, wrong one. For procedure 4, we're doing something similar, but we're not inoculating Serratia marcens into a broth. Instead, we're inoculating Serratia marcens into a slant. So procedure 4, you use aseptic technique to take Serratia marcens out of a stock solution and then streak them in an S-like motion on the slant. A solid growth medium is useful because we can more easily detect contamination. For example, Serratia marcens we expect to be red wherever we streaked it. But if it's not red in any location, we know we have a contaminant. And we can detect even minor contamination. Doesn't have to be major. All right, this tells you the procedure 4. I'll let you read through it. But if the slant does not show confluent or colony growth, then the conclusion is no organisms are growing on the slant and aseptic transfer was unsuccessful. Here we have the picture for procedure 4. And then here are the questions for procedure 4. The first is the bacterial growth on the slant. State yes or no. How do you know? State how you know. And then does it appear to be the bacterium you inoculated or a contaminant? And then state what it is. You should tell why, although I'm not going to require that. All right, any questions about this lab? All right, there's the worksheet to the lab. And I will try and make a change here. So start working on the lab and I will be here at least until 8 o'clock to answer any questions. All right, any questions? If not, I'm going to stop the Hello, there was a question about the first quiz, the practice quiz. And they wanted to know what it covers. Oh, I don't really say. It does cover Lab 00, and it does cover Chapter 1, but not Chapter 1 that we covered today. So it'll be early in Chapter 1. And that's all it covers. Chapter 1, the practice quiz, and Lab 00. Oh, it does cover the syllabus as well. Like I gave you a number of procedures we follow in the lab. So it'll cover that as well. All right, any questions about that?
Oh, that's good. Thank you.
Okay.